Hi everybody and welcome back to this short course of Bioinorganic chemistry. We have been discussing our life with dioxygen. Most organisms require molecular oxygen in order to survive in this earth. We are going to discuss about oxygen carrying proteins in biology. As you all know that the oxygen is the source of all energy. We take oxygen from the environment and this oxygen goes through the blood and responsible to generate energy, water and carbon dioxide. And then also this carbon dioxide has to brought back and released to the environment. So, this is what is happening in our body. So, inhale oxygen and we release carbon dioxides. We will now discuss that how this oxygen is getting circulated or getting transported in our body, whereas how this carbon dioxide also getting released. Now, before going to discuss, let us look at what are the dioxin carrying proteins are available in biology. So, these are all tabulated over here, these are all called metalloproteins like hemoglobin and myoglobin. Their active site structure of deoxyhemoglobin and myoglobin contains iron to heme center. However, after oxygen binding, they convert to oxy form, red in color and iron 2 converted to iron 3. We will discuss in details. Now, hemoglobin, that the molecular weight is very large because of its tetrameric nature, 64,000 Dalton, and higher animals have this hemoglobin in their body. Myoglobin also heme protein and the color change from deoxy to oxy is purple to red. The molecular weight is around 17,100 Dalton. Higher animals have this myoglobin in their body. Now, in contrast, Hemerythrin is also iron containing protein, but non-heme proteins, and in deoxy form it is iron 2 diiron proteins. We will discuss in details. Now, color change from deoxy to oxy, colorless to burgundy, molecular weight is very high 108,000 and invertebrates have hemerythrin in their body. Hemocyanin, however, is a copper containing proteins. So, 2 copper center is present here, we will discuss in details, and color changes from colorless to blue, from deoxy to oxy form, and having a very large molecular weight around 9 into 10 to the power 6 Dalton. Arthropods have hemocyanin in their body. So, let us first talk about the hemocyanin. Now, hemocyanin, it is an oxygen carrying protein found in mollusca and arthropods. The asymmetric subunit mass of around 75 kilodalton forms chains as large as 460 kilodaltons for 6 subunits. Hemocyanin functions as oxygen transport molecule and ensures bodily tissues have those slow movers have adequate dioxygen. This is a schematic representation of hemocyanin, deoxy and oxy, and you can see these structures, excess structure of deoxyhemocyanin and oxyhemocyanin. You see dioxygen bridges between 2 copper center and copper 1 converts to copper 2, it is a oxidation and dioxygen getting reduced to peroxides. So, metal getting oxidized and oxygen getting reduced and they are reversible. Now, deoxyhemocyanin active site structure is shown over here. As you can see that copper ion surrounded by 3 histidine moiety, and the internuclear copper copper distance is around 3.7 plus minus 0.3 angstrom. Copper nitrogen distances are nearly around 2 angstrom. This is square planar geometry around copper, and there is an empty cavity sites available for possible dioxygen binding. This is the X-ray structure of deoxyhemocyanin. As you can see that there is a huge protein chains wrapping the dicopper units, which are sitting at the center, and once you remove this protein chain, you can see that clearly to copper unit they are separated around 3.7 angstrom distance, and as I have just said that this 3 histidine group coordinated to the copper center. So, there is a empty site where dioxygen possibly can bind. Let us see what happens. In oxyhemocyanin, the color change from colorless to blue color because it is copper 2. So, dioxin binds and forms a bridge between two copper 1 atoms, oxidizing them to copper 2 and dioxin getting reduced to O2 minus. The internuclear copper copper distance was decreased from deoxy. It was 3.7 angstrom, now it become 3.6 angstrom, and square planar geometry is compromised around copper. This binding distorts coordination of the protein around histidine bound to copper ions, and two copper 2 ions are very strongly antiferromagnetically coupled at room temperature. So, this is the protein structure of oxyhemocyanin. As you see that again the huge protein chains wrapping this molecule, however, dioxygen comes and binds between these two copper center, which you can see very clearly. And if I remove these huge protein chains, then you see that inorganic molecules where this dioxygen binds in O2 minus, this dioxin is bridging between these two copper unit and bringing two centers close enough, so that it can properly binds dioxygen. Now, this is the schematic presentation between deoxy and oxy. As I have said that the color is getting changed from colorless to blue from deoxy to oxy, and copper 1 getting oxidized to copper 2, whereas dioxygen getting reduced to O2 minus, that means peroxides. How we know that this is peroxides? Because resonance Raman stretch of OO is around 803 centimeter inverse, which confirms the peroxo linkage. Also, these two copper center in oxyhemocyanin are strongly antiferromagnetically coupled, whereas these two copper ion here in deoxy, their copper 1 is a D10 system. So, it is diamagnetic. Now, this is what is the spectral change taking place upon oxygenation to hemocyanin. So, these dotted lines are deoxyhemocyanin, as you can see over here. However, after oxygenation there is a huge change in the absorption spectroscopy, and you see that there is a band around 5-8 nanometer, and this is because of the considerable charge transfer between the coordinated peroxo group and the metal center. So, this confirms again the binding of peroxo within these two copper 2 centers. Now, many people have tried to model this hemocyanin and try to understand that how actually dioxygen binds reversibly, and in order to confirm that they have used different tridentate ligands. I am showing one of such examples having a pyrazole borate ligand. You see this tridentate pyrazole borate, and which actually resembles to 3-histidine group, and this binds dioxygen, and this is the excess structure shown over here. This is a schematic representation. What is being seen that yes, this is the copper 2 center, and also the resonance Raman O stretching frequency is around 741 centimeter inverse, further confirming the peroxo linkage. So, this is further validated by the model study, which confirms that the dioxygen is in O2 2 minus in oxyhemocyanin. We will talk about Hemerythrin, another oxygen carrying proteins in biology. Now, this is the octameric structure. Hemerythrin octamer of monomeric subunit of around 13.5 kilodalton, and as I have said already that it has been present also in many organisms. Now, this is a schematic representation between deoxy and oxyhemerythrin. So, in the deoxyhemerythrin, here again this is the diiron center. It is a non-heme proteins where there is 2 iron center having iron 2 state, and there are one aspartate and one glutamate bridges between these 2 iron center, and also a hydroxo group bridges between these 2 iron. In H iron there are 3 histidine moieties in iron 1. However, remarkably in iron 2 it is only 2 histidine. So, iron 1 center is 6 coordinated, whereas iron 2 center is 5 coordinated. That means, there is a vacancy in the coordination site where dioxygen can possibly bind. This indeed happens, and as you can see in oxyhemerythrin, the dioxygen comes and binds over there, and I will show you soon this dioxygen again converts to O2 2 minus, that means peroxides, and iron 2 converts to iron 3. So, metal is getting oxidized and dioxygen getting reduced. Okay. The excess structure is shown over here. As you can see now, this is in case of oxyhemerythrin, it becomes oxo-bridged. Indeed, it is converted from mu hydroxo to mu oxo. This is also a remarkable change in the structure and magnetic properties. We will see all this in details. So, this is what is deoxyhemerythrin. Let us look very closely. As I have said that 3 histidine moiety is ligated to the iron 1. In contrast, the iron 2 only 2 histidine moiety binds to that, and this is aspartate and glutamate bridging between these 2 iron centers, and both the centers are iron 2. And also there is a hydroxo group which bridges between these 2 iron center. Now, this types of asymmetric coordination environment is possible in biology in presence of protein. If you like to make similar molecule in the laboratory, it would be extremely difficult. I will come into little bit more details about that. However, interestingly, as I have already said, that iron 1 center is 6 coordinated, whereas iron 2 center is 5 coordinated, where dioxin actually comes and binds. So, this kind of situation, this asymmetric coordination environment is stabilized due to the huge protein chains around this molecule. This is a schematic presentation where deoxyhemerythrin converted to oxyhemerythrin in presence of oxygen, and oxyhemerythrin goes back to deoxy, releasing the dioxygen. So, they are reversibly interconverting between each other. And as I have said that this iron 2 center binds dioxygen, and this dioxygen is no more in neutral form, it O2 converted to O2 2 minus, and you see that this proton is there, which is actually hydrogen bonded to the mu oxo group, which is present in oxyhemerythrin. Also, this results from deoxy to oxy a huge color change from colorless to purple, and iron 2 center, which is high spin in the deoxy state, becomes low spin, and also iron 3. So, this peroxo coordination in oxyhemerythrin is further confirmed using the resonance Raman stretching frequency of dioxygen, which gives a value of 844 centimeter inverse, and thus confirming the peroxo linkage between two diiron center in oxyhemerythrin. You can see that in deoxy a hydroxo group which bridges between these two diiron centers, whereas in case of oxy this hydroxo converted to just an oxo group which bridges between these two, we call it mu oxo. So, mu hydroxo converted to mu oxo. Now, how we know that? And the magnetic study indeed confirms that yes, this is mu hydroxo getting converted to oxo, because if it is mu hydroxo, then the coupling between two iron center would be less. This indeed happens, that antiferromagnetic coupling, of course, two iron center undergo antiferromagnetic coupling, and the value is much less, like minus 10 centimeter inverse in case of deoxy state, whereas in case of oxyhemerythrin the values has changed remarkably. This coupling, two iron center undergo antiferromagnetic coupling, and the values is much larger, minus 77 centimeter inverse. So, this coupling is much stronger, and this also confirms that yes, this is indeed mu oxo, because we have seen in model study that if this is converting from mu hydroxo to oxo, there is a huge change in the coupling constant values also. Here I am showing the spectral change upon oxygenation of hemerythrin. So, you see that met hemerythrin means oxidized, means in iron 3 form, however, without dioxin coordination. In oxyhemerythrin, it is also iron 3, but with oxygen bound form. Of course, oxygen bound in O2 2 minus. So, this gives a remarkable shift of its UV visible spectrum. As you can see, there is a band around 500 nanometer, which is due to a considerable charge transfer between the coordinated peroxo group and the metal center. These also suggest that dioxin binds to this iron center. X-ray structure of this oxyhemerythrin is shown over here. As you have seen in case of hemocyanin, there is a huge protein chains are wrapping around this diheme center, and the diheme unit sitting at the center where actually dioxygen comes and binds. So, let us remove this protein chains and see that how actually dioxygen binds. You can see that dioxygen binds like this in O2 minus form, and 2 iron center is bridged by a mu oxo group. As you have seen that glutamate and aspartate are bridging between these 2 iron center, and this 3 histidine group for one iron center, whereas another iron center coordinated to 2 histidine, which we have just looked at in the previous slides. So, X-ray structure also confirms that all this coordination environment along with their angles, distance, and we now know exactly how dioxygen binds to which center, what is the geometry. If you have a protein structure, and then you know all details of that. Now, if you try to model this hemerythrin in the laboratory, lots of people around the globe has tried to model this hemerythrin, and here I have given two very famous example. As you can see that this iron center is no longer asymmetric, they are symmetric now. It is impossible to make asymmetric iron center, which we have observed in case of hemerythrin without protein, okay? Because protein makes them possible two different iron centers. This 3 histidine group is replaced by pyrazoyl borate, a tridentate chelating agent, and it is in the met hemerythrin means it is in iron 3 state, and you can see that 2 iron center is bridged through the oxo group. And there is another examples also when the tridentate ligand is a different triazocyclonone, then also it was reproduced this made hemerythrin, this it also mu oxo bridged between 2 iron center. So, interestingly these complexes have no vacant site for dioxygen binding, and the synthesis exemplify the spontaneous self assembly. So, it is very easy to make this kind of molecule spontaneously, they form this diiron unit. However, since there is no vacant site around iron, so dioxygen cannot bind, and the synthetic model could not reproduce the two different iron center, one is 6 coordinated, another is 5 coordinated. So, that dioxygen comes and binds in at the 5 coordinated site, and which possibly mimic the hemerythrin as observed in biology. So, we have been discussing various dioxygen carrying proteins in biology. Today, we have discussed two such oxygen carrying proteins, hemocyanin and hemerythrin, in great details. They contain dicopper and diiron centers in their active sites. Once dioxin binds to the dimetallic core, metal centers get oxidized by 1 unit H, while dioxin gets reduced by 2 electrons to form peroxide. Also, two metal centers undergo strong antiferromagnetically coupled in their oxyform. I also have highlighted how the beautiful design principle adopted by our mother nature control reversible binding of dioxygen in these proteins. In my next lecture, however, I will talk about the dioxin transport and storage in our own body. Nature has designed hemoglobin and myoglobin. Hemoglobin selected for transporting oxygen, while myoglobin stores oxygen. I will discuss how protein chains are actually responsible for reversible dioxin binding in hemoglobin and myoglobin. I will also highlight how the beautiful design principle makes all these processes possible in sustaining our life. Thank you.